Hello, my name is Andrew Brummett, and I am a senior at Indiana University, Indianapolis. I will be presenting on the topic of volition and homosexuality. Volition is also called choice and concerns whether or not something is voluntary. So, if homosexuality has volition or has a nature of volition, then it is a voluntary process that is chosen. If volition is not present, then homosexuality is an involuntary process that one has no control over. This presentation will dive into several ways that volition can be researched, and what those research methods have yielded in the literature by examining several key studies. So, what is volition and why is it important? Before diving into research methods, we should establish why volition is important. Why should we concern ourselves over this topic, and why is it researched so frequently? Unfortunately, homophobia is still a large issue in the 21st century and can have damaging and even life-threatening effects on queer populations of all backgrounds and countries. A large foundation of homophobia is the thought that homosexuality or other forms of queerness are a choice and can be controlled. When people think this way, it is much easier to harbor negative feelings toward homosexualities. This study, titled Belief in the Free Choice Model of Homosexuality, a Correlate of Homophobia in Registered Nurses, first authored by Blackwell, was performed in the United States on nurses and examined attitudes surrounding free choice in homosexuality. The study found that there was a strong positive correlation represented by the asterisk critical ratio value between nurses believing that homosexuality was a conscious or voluntary lifestyle choice and harboring homophobic attitudes. So in other words, this factor was found to be the strongest predictor of nurses harboring homophobic attitudes. Otherwise worded as, if someone has a belief in the free choice model of homosexuality, then they are likely to believe that homosexuality was a conscious lifestyle and had the highest levels of homophobia. So it is important before we dive into other research methods that in the literature generally, when people talk about queerness, they talk about it in a binary between either heterosexual or homosexual. Many do not make a delineation between bisexual. So when anyone says homosexual, it means any sexuality that deviates from heterosexual or is sexually deviant, which may be an offensive term, but it is used in research quite often still. So how can we study the human bee brain and body to establish the presence or absence of volition in homosexuality? The first modality, moving from left to right, is to use basic MRI studies to evaluate the structure of the human brain. Basic MRIs, or magnetic resonance imaging, can be used to identify narrow anatomical differences between control and treatment groups, which in our case is heterosexuals versus homosexuals and other forms of queerness. Next, we can use fMRIs to identify differences in brain activity in response to external and internal sexual stimuli. Please note that the F in fMRI stands for functional. When brain regions become active, they create an increased oxygen demand and therefore receive increased blood flow to account for increased activity. This activity can be traced in an image called a blot scan, and this increased blood flow can be detected using this fMRI scan and artificially colored to create an activity representation, which is then overlaid with a simultaneously imaged MRI scan. The next modality is genetic screening and testing, which has been made easier with the modern presence of large genome banks from sources such as 23andMe and other family tree tracking services. This can be used to detect genetic differences between heterosexuals and queer individuals. The last modality is twin studies, which aims to detect the genetic influence of certain traits by examining how they are conserved amongst different types of twins. So when you do a twin study, you use either identical, fraternal, and normal siblings. If a genetic trait has a higher amount of concordance between twins, identical twins, and fraternal twins, it suggests that the trait is more influenced by genetic factors. Therefore, it is less malicious and is more innate in your nature. What has neuroanatomical MRI research found on this topic? Well, this is a series of studies that concerns MRI findings that show neuroanatomical differences between heterosexuals and homosexuals. The first is first authored by LeVay and showed that interstitial nuclei of the anterior hypothalamus zone 3 was twice as large in heterosexual men than homosexual men, which is a very long way of saying that the hypothalamus in heterosexual men was found to be much larger than in homosexual men. And this paper was titled A Difference in Hypothalamic Structure Between Heterosexual and Homosexual Men. The next study, which is first authored by Vodanov, is titled Brain Structure Changes Associated with Sexual Orientation, and showed that heterosexual men had larger cell masses in the precentral gyrus, which is imaged here, and the thalamus, and also the puny men than homosexual men. So with this, they were also able to draw a strong association between the sizes of these structures and whether or not an individual would be heterosexual or homosexual. In the next study, which is first authored by Manzori, found that homosexual men, and it's titled Multimodal MRI suggests that males with homosexuality may be linked to cerebral midline structures, show that homosexual men have a much larger and thicker anterior cingulate cortex, precuneus, and occipital temporal cortex than control heterosexual groups. So all these studies suggest that there are innate neuroanatomical differences between homosexual and heterosexual men, whether that be larger or smaller structures. They can also show that they're all over the brain. So you can see here that we have hypothalamic structures, we have precentral gyro structures, which are in the frontal lobe, and we have putamen structures, which are in the insular cortex, along with occipital temporal cortex, which is very highly involved in vision, and then the anterior cingulate cortex, which is involved in emotions and memory processing. So these are very strong evidence that these changes are all over the brain and are very widespread in our nature. So what has fMRI research shown in terms of this topic? So this paper, which is published in the Journal of Human Brain Mapping, is titled Brain Response to Visual Sexual Stimuli in Heterosexual and Homosexual Males, and was authored by Paul. So keep in mind that fMRI can be used to study our responses to external sexual stimuli and the differences in those responses amongst treatment groups. So this journal aims to describe differences in the activation of brain regions such as the insular cortex and hypothalamus between heterosexuals and homosexuals. First, the study establishes that heterosexuals feel greater sexual arousal to the study's examples of heterosexual stimuli than do the homosexual stimuli, and vice versa in the homosexual group. So you can see here that in heterosexual individuals, that a significantly higher response in sexual arousal rating in response to the heterosexual stimuli in white than the homosexual stimuli in gray. And this is flipped right here for the homosexual individuals. So gray much higher, white much lower, which establishes the validity of the heterosexual homosexual stimuli. So in the methods of the paper, if you decide to read it, the heterosexual stimuli, what they mean by that is heterosexual appealing pornography, and then homosexual stimuli was homosexual appealing pornography. So this was necessary as to establish the validity of the sexual stimuli. You can see here that there's increased. So here's the first fMRI study. So for A, this is a heterosexual watching homosexual porn, and you can see that right here in this circled region, which is the insular cortex, there's very little activation. But when that same heterosexual watches heterosexual pornography, the insular cortex is lit up with action. So you can see that a very higher neuroanatomical difference is present here when viewing the appropriate sexual stimuli. There's also proof of concept in the study that the occipital cortex is lighting up when viewing visual stimuli. So occipital cortex obviously does our visual stimulus processing. So this shows that the fMRI scan is actually working. And then for B and D, this is a homosexual watching heterosexual pornography, and you can see very little activation in the insular cortex. Whereas for D, this same homosexual is watching homosexual pornography, and there's much higher activation in the insular cortex. And again, you can see here very uniform activation in the occipital cortex between the two treatments. So you can see that the fMRI scan is working. And then for A and E, this is a homosexual watching homosexual pornography, and the hypothalamus is being studied. So you can see the homosexual has the hypothalamus is lining up in response to homosexual stimuli, and then they're watching heterosexual stimuli with very little stimulus. And then for B, this is a heterosexual watching heterosexual stimuli with activation of the hypothalamus, and then the same action for the homosexual, so very little activation when watching the inconcordant pornography. So this study really shows in great detail that we have innate responses to outward sexual stimuli that we cannot control, because these are neuroanatomical differences with different responses to stimuli. This is not something you can control, and you cannot make a choice in your fMRI scan reading. So this study, first authored by Ghana, shows that some genetic associations of homosexuality and sexual behavior. So this paper is titled Large-Scale Genetic Genome-Wide Association Study Reveals Insights into the Genetic Architecture of Same-Sex Sexual Behavior, and was authored by Ghana. So the first image on the left is a screening of all 23 pairs of chromosomes, with 5 loci identified with strong possible associations with sexual behavior. So these genetic loci were then used to perform a genetic correlation analysis by questionnaire on subjects. The study found a strong genetic correlation, most interestingly between to whom are you sexually attracted to, and how do you label, identify, and think about yourself, and whom do you have sexual fantasies about. So that's very interesting because to whom you are sexually attracted to has a strong genetic association with how you identify with yourself and how you think and label yourself, and also how you have uncontrollable sexual fantasies about. I find this interesting because they highlight our intrinsic, how intrinsic our sexuality is to our overall personality, and in my opinion, kind of in a way, uh, validates the presence of volition. Although this study did not find a quote-unquote gay gene, they were also able to find these 5 loci that were highly suggestible for a genome-wide association study. So keep in mind that people were asked to fill out a questionnaire with their genomes available, and this association study was also ran backwards to find these loci again. So it's a very powerful way to associate homosexuality or any type of sexuality with your genetic makeup. Have twin studies shown on this topic. So this study, titled Homosexual Orientation in Twins, it reported on 61 pairs and 3 triplet sets, published in the Archives of Sexual Behavior, and first authored by Whitham. And I apologize, this is the copy and paste an abstract here, but they had no figures. So in this study, authored by Whitham, the genetic basis of homosexuality is further explored through twin study. So twin studies, again, examine the concordance of traits in identical twins, fraternal twins, and normal siblings. Concordance is measured in rates and describes the amount of times that a trait is present in both twins. So if a trait has a high concordance in identical twins, then lower concordance in fraternal twins, and then an even lower concordance in normal siblings, this suggests that the trait is genetically influenced. In this study, 38 pairs of identical twins had a concordance rate of 65.8% for homosexual orientation. Fraternal twins had a concordance rate of 30.4%. These findings suggest that homosexuality is genetically influenced, and as the concordance rate drops as the genetic relationship between the twins weakens from identical to fraternal. This study was weakened by the fact that it utilized a questionnaire with no objective measurements of homosexuality and had small group sizes. It still, however, possesses a very interesting finding that suggests a genetic makeup between homosexuality and any other sexuality. What can we conclude? In my opinion, we conclude that MRI structural research has found evidence of neuroanatomical differences between heterosexual and homosexual men and women, which may suggest that there are innate neurological processes that erase volition. We also conclude that fMRI research has found evidence that homosexual and heterosexual people respond to sexual stimuli in different ways, that suggests that responses to sexual stimuli are involuntary and therefore violate the idea of volition. We can also find that genetic studies have not found a gay gene, but have found strong genetic associations between certain sexual behaviors, with certain genetic foci possibly identified as being responsible. We also find that twin studies have found that homosexuality has a high genetic accordance, with sexual preference being conserved among identical twins more often than fraternal twins.